Rahim. Now we will discuss lecture number 5 of chapter number 5, structure of seed and seed germination. The key concept that we will discuss in this lecture: structure of seed germination, condition necessary for seed germination, condition necessary for germination of seeds. First, we will discuss the structure of seed. A seed, also known as mature ovule, is a miniature. Miniature means small plant with protective cover in a suspended state of development. Suspended state means that when we don't dig or keep the seed in the soil, so during this time the seed don't germinate. So such condition starts suspended state. Following are the major component of seed. First, seed coat. Seed consists of seed coat or testa. It is a black brown in color and cover the embryo. Beneath the testa or inside the testa is a thin coat, the tegmen. Tegmen is the thin coat present behind the testa, which is fused with the testa. Hilum. Hilum, there is a scar on seed coat called hilum. It is where the seed is attached to the ovary wall. Through scar, the seed attached to the ovary wall or fruit wall. Micropyle. At one end of hilum there is micropyle. This is the same opening through which the pollen tube enters ovule. So, micropyle are used for the entrance of pollen grain to ovule. Seed uses it for the absorption of water. So, it is a dual function. It provides a channel for the entrance of pollen grain, and similarly, it also used for the absorption of water. Embryo is usually an immature plant. It consists of a radical, plumule, and one or two cotyledon. So the embryo consists of three parts. One is the radical, which produce the future root. One is plumule, which produce the future shoot, and two cotyledon, which produce the future leaves. Or seed leaves. The food also stored in the cotyledon. The radical of the embryo develop into new root, while the plumule develop into shoot. Epicotyle. The embryonic stem above the point attachment of the cotyledon is called epicotyle. Hypocotyle. The embryonic stem below the point of attachment is a hypocotyle. Within seed there is a store of nutrients for the seedling that will grow from embryo. Seedling is a small plant. When the seed germinate or the embryo germinate, so it produces small plant. This is called seedling. In angiosperm, the stored food is derived from the endosperm tissue. The endosperm tissue are present in the cotyledon. This tissue is rich in oil or starch and protein. So these three components are present in the endosperm: protein, starch, and oil. Cotyledon. In many seeds, the food of the endosperm is absorbed by cotyledon. This is the structure of seed, and this is embryo, and the first part, the outer part, is called the seed coat, and these two parts are called the cotyledon, and this is the plumule. The upper part is the epicotyle, and the lower part is called the hypocotyle. This is the radical, and this is the dicot seed. What are the dicot seed? The seed having two cotyledon are called dicot seed. Di means two, cot seed means cotyledons. For examples, beans and peanut, etc. This is monocot, for example, maize and wheat. So these are the monocotyledon seeds. What are the monocotyledon seeds? The seed having one cotyledon are called monocot seed. This is the cotyledon, only one cotyledon present in the monocot seed, and this is plumule. So this is all about the structure of seed. Now the second part of this lecture, types of seed germination. Seed germination is basically, but before discussing the types of seed germination, first we will define the seed germination. In the dry seed, the embryo is alive but inactive. When it is placed in the soil under suitable condition, the embryo becomes active and grow into seedling. Suitable condition means the favorable environment for seed germination, in sense of suitable temperature, suitable soil, and climate. The process by which the inactive embryo in the seed become active and develop into seedling or small plant is called seed germination. So seed germination is the process in which the embryo is active and the embryo grow and convert into small plant or seedling. This process is called seed germination. Types of seed germination. Germination in plants is of two types. Epigeal germination. The type of germination in which the cotyledons are forced above the ground. Hypocotyle elongates and cotyledons become photosynthetic. So in this case, the hypocotyle elongates. Due to the elongation of hypocotyle, the cotyledon push above the ground. The seed coat also emerge from the soil. For example, seed of melons, cucumbers, and beans show epigeal germination. The diagrams show epigeal germination. Stage 1, this is bean seed keep in soil. Stage 2, the radicle come out. And in stage 3, the hypocotyle elongate. So this is the hypocotyle, and when the hypocotyle elongate, the radicle also grow inside the soil, and as a result, the cotyledon push upwards. In stage 4, the cotyledon push upward above the soil, and this is the epicotyle stage. In stage 5, complete seedling develop. So this is the type of epigeal germination in which the hypocotyle elongate and push the cotyledon above the soil during germination of the seed. So this is epigeal germination. Hypogeal germination. The type of germination in which the cotyledon remain in the seed below the ground. Epicotyle elongate and cotyledon do not become photosynthetic. Photosynthetic means that they remain inside the soil during the germination, and the leaves do not develop from the cotyledon, so it remain non-photosynthetic. Seed coat remain in the soil of ground seed and may show this type of germination. So in this case, the epicotyle elongate and push the radicle inside the soil, and the cotyledon remain inside the soil, become non-photosynthetic. So this is a hypogeal germination. The diagram show the hypogeal germination. In stage 1, the seed buried in the soil. This is the radicle, this is the hypocotyle, this is the epicotyle, and this is the hypocotyle. So the epicotyle elongate. The epicotyle elongate, and this is the cotyledon. The cotyledon remain inside the soil, become non-photosynthetic, and the radicle elongate. And in stage 4, root developed, and epicotyle further elongate, develop shoot. And this is the small seedling. The cotyledon remain inside the soil, become non-photosynthetic, and this is the hypocotyle and radicle. So this diagram show the hypogeal germination. Condition for seed germination. The most important condition for seed germination include water, oxygen, and favorable temperature. First, we will discuss water or moisture. Seed of most plants have low water, and germination cannot occur until seed coat or other tissue have imbibed or taken water. So when the seed coat absorb water and there is a sufficient amount of water in the soil, so then the seed will germinate. If there is a low quantity of water in the soil and the seed coat don't get soaked in the water or absorb water, so it will become inactive stage or in suspended state, and the seed will not germinate. The absorbed water is used in the digestion of stored food, and it is also in the elongation of hypocotyle and epicotyle. So due to epicotyle elongation, shoots develop, and due to hypocotyle elongation, the root develop. Oxygen. Oxygen is essential for the respiration in the cell of embryo. So for the germination of embryo, food is required. The stored food require oxygenation for respiration. So oxygen is required for the oxidation process, and respiration occur, energy will release, and the embryo will germinate. Temperature. Seed differ greatly in their temperature requirements for germination. So some seed required low temperature. For example, the wheat seed required low temperature to germinate. For example, seed of the wheat germinate in winter season because due to the presence of low temperature in the environment. Similarly, other seed that required high temperature, they are grow in the spring season and summer season. The optimum temperature for the germination of seed of most plant range from 20 to 30 degrees Celsius. Above this temperature, the food and the enzyme become denatured. The enzyme become inactivate. They digest the food, and the seed will remain dormant. So this is all about the structure of seed, seed germination, its type, and condition for seed germination. So if you have any question, comment me. I will give answer. Thank you.